Hello class, this is my third video. I started because the other one a while ago did not have any sound. So let's go ahead and pick back up. We are looking at causes and development of infection in the body. So first off, you have to have the pathogen, right? And it has to be able to enter the host, either through a mucous membrane, broken barrier, or in the respiratory, nasal area, however you want to get in it, but it has to get inside. Then the pathogen has to reach the a site that is conducive to its growth. So once it's inside, it's still not good enough. It's got to get it has to get to a site that's conducive to its growth. And depending, it's usually maybe an inflamed area or very low oxygen area or high oxygen area. So it just all depends. And then once it gets to its desired area, it will begin rapidly reproducing. And the first thing you might see is some prodromal effects, which is general sickness effects. Not very non-specific. Okay, you might not even have very many signs at all. Then, as it increases even more, we get some of those acute signs, and those acute signs are going to usually indicate what the disease is, and we can use some other things along with those signs for diagnosis. Then it can take one or two tracks. It can continue to overwhelm the immune system and antibiotics and all those types of things, and that would lead to septicemia and death, which is sepsis of the blood by the bacteria, so by the toxins and so on and so forth. Or we can begin to overcome the disease, and we're kind of just fighting that battle. So how do we do that? We use the host defenses. Nutrients supply can be decreased. Waste and cell debris is kind of increased, where we're doing all that cell damage, and we're going to be phagocytosing like crazy. And then we can use some antibacterial drugs that will sort of kind of lower. And then as we sort of kind of overcome disease, recovery begins to occur, and either we will totally wipe out the infection, or we could always just leave small pockets in our body and it could continue on. It will be it might have very mild signs, but it can be very destructive and have detrimental long-term effects. Signs and symptoms: inflammation. We talked about it in depth. Pain, swelling, redness, and warmth. Typically, if it's a bacterial infection, it can produce a purulent exudate. If it's a viral infection, it typically produces a clear, serious exudate. So just depending on basically what that fluid is that could be excreted out of that area. Systemic signs: fever, infection, fever. They go hand in hand. Fatigue and weakness. Think about getting the flu. Your fatigue, weakness, headache, and also nausea. Nausea is just a systematic sign too, with all kinds of different infections, and it'd be bacterial or viral, fungal, or whatever. We sort of kind of see some of that. Methods of diagnosis. The primary method we use now is culture samples. We will culture that specimen out of the site of the infection, and we send it off to the lab, and the lab will try to grow it. And that's what it means by culture. It tries to grow it, and we can see whether it grows on this plate with this type of antibody, this plate with this type of antibody, and see what it's sensitive to. We can also stain and the bacteria, see if it's positive or negative, and that can determine antibiotic treatment. We can do blood tests, which look at our white blood cell count, and by looking to see whether it's really high or really low, it can indicate whether it's a bacterial or viral infection. So if it's really high, we call that leukocytosis, and that means that it's a bacterial infection. Bacterial infections usually cause lots of white blood cells to be made and grow. If it's a viral infection, that can sort of kind of lower the white blood cell count, and it can cause leukopenia. And at the same time we're doing this white blood cell count, looking at the total number, and then we're also going to look at the differential number. So we can look at the neutrophils, eosinophils, basophils, monocytes, lymphocytes, and see which ones have the most, and that can indicate what type of infection it is too. The role of antibiotics or antimicrobials, which I should say there is a difference. Antibiotics are naturally synthesized from organisms, such as our bread fungus producing the penicillin. That's an antibiotic. Antimicrobials mean that they will attack or made. They can be synthesized also. But they both kind of function the same. They both are going to do different. Have a bactericidal effect. Or bacterial inhibiting effect. So one thing is we can do is inhibit and interfere with bacterial cell wall synthesis. So we interfere with the cell wall, and therefore it can't divide and it can't make new cell wall. And a drug that does that is penicillin. It works directly on the cell wall. We can increase the permeability, and we all know we have to maintain homeostasis of our body. A bacteria has to maintain homeostasis also. So we can increase the permeability of a bacterial cell membrane, and that can cause it to die or slow it down. And polymyxin can do that. We can interfere with the protein synthesis, which is the proteins the bacteria needs to make to live. And bacteria's protein synthesis is very similar, is similar, but because they use ribosomes, but their ribosomes are different than our ribosomes. So we can attack those ribosomes, and therefore they cannot make any protein proteins or inhibit their protein synthesis. And we use tetracycline for that. And then we can also interfere with their metabolites. So basically, it'd be like us interfering with our vitamins. If we don't have our vitamins and our metabolites from the vitamins, we cannot function. So we can interfere with the bacteria's metabolites. And we use pseudoadenines for those. Amides. Antivirals. Since viruses really aren't alive, what we try to do is slow down their reproduction, because that's really all a virus can do, is it reproduces and produces more viruses. So what we try to do is prevent it from doing this. How can we do that? We can block entry into the host cell. That's number one. We try to block entry by either blocking the receptors that the virus binds to or blocking the process. We try to inhibit the proteins made by the virus. So we can try to inhibit it from being made. We also can inhibit the assembly of the virus. So how does it get put together? We try to stop those chains. So that's what antivirals do. And typically, like with HIV patient, we will give them a cocktail that does several different things in several of these different parts. One might try to block the cell entry of the virus. The other one might try to block the inhibit gene expression. So we're going to try to slow down these viruses. And then antifungals, since fungi are very similar they're eukaryotic and similar to human cells, we have to be kind of careful about how we treat these. We typically try to interfere with mitosis of the fungi, and some of them may increase fungal membrane permeability. Most antifungal agents are administered topically, to the area where the fungus is at. We don't want to be ingesting this stuff because fungi are eukaryotic, and so what's toxic to them is often toxic to us. So we have strict medical supervision for fungi treatment. Then we have antiprotozoal agents, which are going to try to kill the protozoa. And the thing about it is, those also are very similar. Those cells are very similar to our own cells because they're eukaryotic. So we have to be very careful treating those, and we have to be able to treat them at different stages of the protozoa life stage. So one life stage it might be susceptible to this, another life stage it might be susceptible to this. So we have to be careful and treat all the stages.